नमस्कार दोस्तों, स्वागत है आपका विज्ञान घर के दूसरे वीडियो में। इस वीडियो में हम डिस्कस करेंगे जामिया मिलिया इस्लामिया एमएससी बायोसाइंस 2020 के आगे के क्वेश्चंस। तो शुरू करते हैं। पहला क्वेश्चन है, द न्यू टेक्नोलॉजी टू प्रोड्यूस इकोनॉमिकली एंड मेडिसिनली इंपॉर्टेंट प्लांट्स ऑन लार्ज स्केल इज। The four options are cutting and layering, cell and tissue culture, genetic engineering, grafting. As we know that in the case of the plant, the cell and tissue culture techniques have been exploited very well, and it has made possible to grow economically and medicinally important plant. Today we can grow selectively obtain genotypes with high production rate irrespective of their geographical boundaries. These plants can be used for the production of alkaloids, terpenoids, flavonoids, dyes, their different pigments, amino acids, and etc. So, the correct answer would be cell and tissue culture. Again, question is, when a leaf cell is made to grow into a complete plant, it exhibits cellular. As we know, the generation of a new cell from a existing plant where it is made to grow into a complete new plant under culture condition is known as totipotency. Generally, the tip of the leaf is being taken, which has the meristematic cells, which is competent enough to grow into a complete plant, and totipotency need to be correct answer. Which of the following is not true scientific nomenclature? There are four facts have been given, and we have to find out which of the following statement is not correct as far as scientific nomenclature is concerned. As we know, the scientific nomenclature which we use today is being given by the Linnaeus in 1753 in his book Species Plantarum, where he used the Latin nomenclature where the two words have been written. This binomial nomenclature where the two words are there are being represented in italics, where the first word represents the genus of a particular species and the last word is represented as a specific epithet. One need to remember that these names are very specific for a given organism or a population of a species, and irrespective of their geographical location, these names are conserved, provided the organism belongs to a unique species. So the correct answer where the statement names vary with the geographical location has to be there, because it is not considered in the binomial nomenclature. Pigments responsible for photo-morphogenesis is Xanthophyll, Chlorophyll, Cytochrome, Phytochrome. The development of plant where the different developmental pattern is being governed by the light spectrum is called as photo-morphogenesis. As you can see in the diagram, there are three types of pigments are there: phytotropin, which controls the phototropism, cryptochromes, which controls the detoliation, and phytochromes, which controls the photoperiodism. Out of four options given here, the phytochrome has to be correct answer, because phytochrome actually controls the photoperiodism. Where the two alternate forms exist for a phytochromes, PR and PFR. PR is an inactive state which gets activated when absorbed red light, which is around 670 nanometer. The active state PFR, it does all the biological function, developmental patterns, and when it receives the far red light or the light which we can't see, then it again becomes inactive state. The alternate circadian rhythm based developmental pattern is being controlled by the phytochrome. So in the given question, phytochrome has to be the correct answer. Yes, it is. A complete haploid plant can be obtained from Carpel, Anther, root tip, apical meristems of a stem. First, we should know why it is the need of the haploid plant. For your information, haploids, it provides a convenient system for the selection of mutations or the induction of mutation with the desired traits. The different types of mutants in different plant species has been developed over the period of time where these characteristic traits are present in them. These haploid plants in many genera of angiosperm can be obtained in vitro through anther or microspore of ovule culture. So in the present case, the answer has to be B, anther. TLC is generally used as a qualitative analytical technique for the first three options are determining the number of compounds in a mixture. That is also correct. In a normal routine chemistry lab, you have seen the students are doing TLC just to know the reaction progress, rate of the reaction, course of the reaction, and also that how many compounds are present in a given mixture. It can also be used for checking the purity of a compound. One should remember that two different compounds might be having same RF value in a given solvent system, but it is almost improbable that two different compounds, two different analytes may have a variety of RF value in different types of solvent system. It is almost practically impossible, and that's where it can be used to characterize a compound, to characterize the purity of a compound in a selective TLC solvent system. So D answer has to be correct, all of the above. Most of the amino acids found in the human bodies are, as we know, it's a very simple question. The amino acid which is present in a human body for general routine metabolism is L-isomers, and sugars are generally D form. This is very conveniently one may memorize. In biological world, most of the time it is the L-isomers which are being used, and if at all D-isomers have been used, it is not for the routine metabolism, rather than to provide a survival edge for an organism. For example, certain marine snakes, they have a poison called counterfan made up of D amino acids, and in the peptidoglycan layer of a gram-positive bacteria, they also incorporate D-isomers so that their cell wall cannot be degraded by the known proteases, which are very unique for the L-isomers rather than D-isomers. Hence, the answer in this present case has to be A, L-isomer. Question is, an example of omega-3 fatty acid is, there are 4 different free fatty acid examples are being given, and it has been asked which one belongs to omega-3 types of fatty acids. One need to remember that omega classification or omega nomenclature of a free fatty acid belongs to unsaturated fatty acids only. It is not for the saturated fatty acids. And in this nomenclature, the terminal CH3 group, which is being given the omega name, just opposite in direction to the functional group COOH group, is being used as a reference. The first double bond, whichever comes in priority from the terminal CH3 group, as said, is being given omega 2, omega 3, omega 4, and so on and so forth. If you see the following table, the first 4 free fatty acids which are being given, they are saturated fatty acids, so there is no option that it could be belong to any omega. While if you see the palmitoleic acid, for example, it is a monounsaturated fatty acid who has only one double bond, and it belongs to omega 7 category. Alpha linolenic acid, which is a polyunsaturated fatty acid having 3 double bonds, it belongs to the omega 3 category. Remember, omega 3 and omega 6 are good to take for health point of view, and in the present case, linolenic acid has to be the correct answer. Hence, C option is correct. Polypeptide chain formation occurs. As you know that polypeptide chain formation occurs at the ribosomes, where all messenger RNA are read in 5' to 3' direction, and one should remember that polypeptide synthesis always being occurring from amino terminus to carboxy terminal, from N terminal to C terminal. Hence, option D has to be the correct answer. A competitive inhibitors. There are 4 options are being given. Which of the following statement justify the character of a competitive inhibitor? As the word coming competitive, that means the inhibitor is going to compete. Compete with whom? A typical competitive inhibitor, it is an analog to the substrate, natural substrate of an enzyme, and that's where it competes for binding at the active site with the natural substrate, because of which the Km value alters while Vmax remain constant. So in the present case, a competitive inhibitor always going to bind at the active site, and it is going to compete with the natural substrate for the binding. Hence option A is correct. In metalloenzymes, metals are. As we know that almost 1/3 of the known enzymes classes is belonging to the metalloenzymes, and in all the metalloenzymes, a typical metal ions, generally the transition metal ions, are present and which are attached with the protein core of the active site through the coordination bond. One of the peculiar thing of metalloenzymes are that at least one of the coordination sphere of these metal ions are not fulfilled. It's a labile which is used to interact and orient the substrate molecule. So in the present case, in metalloenzymes, metals are attached to the enzyme through coordination bond, which is A, and hence option A is correct. अगला क्वेश्चन है, ग्लूकोज एब्जॉर्प्शन इन इंटेस्टाइन रिक्वायर्स. 4 ऑप्शंस दे रखे हैं. As you know that there are 3 monosaccharides which are being used and absorbed in the intestines. They are galactose, glucose and fructose. And obviously glucose is much more important, and which exploit a sodium glucose symporter for the absorption. What happens is, in the intestinal lumen there is a plenty of sodium ions are there, which as per their concentration gradient they enters into the epithelial cells of the intestine, and along with their movement they drag glucose along with it, and glucose also uses the same symporter or carrier molecule in order to enter the enterosomes. From here the buildup of sodium need to be released, and that's where the sodium is being released into the blood vessels, and by using the sodium potassium ATPase, which uses the ATP energy, and the concentration gradient is still maintained, while glucose is using a uniporter molecule to get released into the blood vessels, from where it is being directed to the liver. So hence in the present case the glucose absorption requires a carrier and sodium molecule, which is carrier is sodium glucose symporter. खाली carrier molecule की भी जरूरत नहीं होगी because sodium is absolutely required. खाली sodium भी use नहीं होगा because carrier molecule is required, and carrier and potassium ये combination भी नहीं बन सकता because carrier and sodium की requirement है. तो option A would be the correct answer. Yes, it is. Ubiquitin is a protein required for. If you see, ubiquitin is a small 76 amino acid long and 8.6 kd long protein which is found in all eukaryotic cells. It is known to perform various functions by conjugating with different proteins. It's one of the most important post-translational modification which is used to keep a check on protein turnover. Once ubiquitin is bind to any protein, it is destined to be degraded, and hence by selectively marking the proteins for degradation, ubiquitin ensure the correct protein homeostasis in a typical eukaryotic cell. Hence in the present case A has to be the correct answer. Lysosomes contains mainly hydrolases, proteases, lipases and cathepsin. As we know that lysosomes are membrane bound vesicles which contains 50 different types of hydrolytic enzymes. All these enzymes are being used to hydrolyze DNA, RNA, proteins and different types of carbohydrates. So as we know that lysosomes do contain proteases, they do contain lipid degrading enzymes, but the most appropriate answer here would be hydrolases because that encompasses all different types of hydrolytic enzymes. So here the option A would be the correct answer. Yes, it is. Inhibitor of replications are required as you know that replication is a very central event to ensure the success of the life, starting from virus to bacteria to even eukaryotes. If we target replication at any stage, we can selectively ensure that a targeted cell will never grow further. And hence, the inhibitors which are selectively targeting either an enzyme or a selective stage of a replication event can be used to control the cancer, to control the proliferation of bacteria or the viruses. Hence, it can be used as an anti-cancer drug, antibacterial agents or antiviral agents. And in the present case, D has to be the correct answer. All of the following statements are correct regarding heme catabolism, except the 4 statements are given, out of which one is not representing the correct different stages of heme catabolism. As you know that RBC has a limited lifespan of 120 days, after which it surrenders to the laws of nature. Into the reticuloendothelial system, of which spleen is also a part, it is being degraded. The protein component globin is being released, while heme component is further being released and catabolized. The heme is worked upon by an enzyme called hemoxygenase, which releases the iron and catabolizes the porphyrin ring as a carbon monoxide. The carbon monoxide is further released and convert heme into a green color compound, biliverdin. This green compound is being further transformed into bilirubin, which is an orange yellow compound and slightly insoluble in nature. From here, it takes its entry into the liver, where it is being made soluble by conjugating a sugar, glucuronic acid, and released as a bile salts into the gallbladder. Into the gallbladder, it's further released into the intestinal lumen, where the microbial flora change this compound into 2 types of further intermediates. One of the intermediate is strucobilin, which is a brown color compound and being originally released into as a component of fecus, and that is where fecus is a slightly brown color in nature. While other component, urobilinogen, is being released, reabsorbed by the liver and released into the kidney, where it is either being again sent for the same journey, or it is being converted into the urobilin, where it is being further released into the urine, and that's where urine has a characteristic color. If you see carefully the all stages of the heme catabolism, only one thing which is objectional here is it occurs in intestine. As you know that heme is basically catabolized up to bilirubin in reticuloendothelial cells, but not in intestine. And that's where the option B has to be the correct answer in the present case. Which of the following has high biological value? First of all, we should know what is biological value. In the protein case, biological value, it represents that how much successfully absorbed protein, the fraction of it is being selectively transformed into the body tissues, and how much is being wasted as an excretion. And if we see that the animal protein, they are having high biological value. In fact, egg, which is supposed to be one of the highly biological value protein, has an index of 100. So in the present case, the B, animal proteins, has to be the correct answer. The necessary condition under which a reaction is at equilibrium is, as we know that any reaction which is supposed to be at equilibrium, their delta G value is 0. Initially, a typical reaction with a delta G value negative, it starts and keep running spontaneously till it achieves equilibrium, where the delta G value goes to the 0, and that's where in the present case the A option has to be the correct answer. आगे चलते हैं. For an exothermic reaction, the increase in temperature will shift the equilibrium towards. चार ऑप्शंस देखते हैं. लो वॉल्यूम साइड, हाई वॉल्यूम साइड, टेम्परेचर हैज नो इफेक्ट. ये तो बिल्कुल गलत होगा, बिकॉज टेम्परेचर इज अ क्वांटिटी which has a great effect over the equilibrium constant, and then D option towards product side. देखने में यहाँ पे चारों ही ऑप्शन गलत लग रहे हैं, क्योंकि एक एक्सोथर्मिक रिएक्शन का मतलब है दैट एनर्जी इज बीइंग रिलीज्ड, एंड अगर आप ऐसी रिएक्शन को टेम्परेचर और देंगे तो एज पर ली चैटलियर प्रिंसिपल द इक्विलिब्रियम विल शिफ्ट टुवर्ड्स द लेफ्ट हैंड साइड. एंडोथर्मिक में ये राइट हैंड साइड पे शिफ्ट होता है, यानी प्रोडक्ट साइड पे शिफ्ट होता, बट हियर इन दिस केस इट विल शिफ्ट टुवर्ड्स द लेफ्ट हैंड साइड, दैट मींस टुवर्ड्स द रिएक्टेंट साइड. तो आंसर सी और डी तो गलत हो गए. नाउ ऑप्शन कम्स ए एंड बी. अब सिंस हियर इट इज रिटर्न लो वॉल्यूम साइड एंड हाई वॉल्यूम साइड, जो कि अपने आप में सफिशिएंट इंफॉर्मेशन नहीं है, तो लेट्स अंडरस्टैंड इन अ डिफरेंट वे. As we know that exothermic reaction, they are spontaneous by releasing the energy, and they forms the laws of thermodynamics, where an ordered organized system is being converted into unorganized system, random system, by increasing the entropy. And that's where from the available options we can consider that low volume side could be more close to appropriate. Rather than saying 100% that it is the correct answer, we would say that it is more appropriate from the options available. Otherwise, if it would have been written towards the reactant side, then that could be the correct answer. तो इस क्वेश्चन में या तो दो चीजें गलत हो सकती हैं, या तो आप एंडोथर्मिक रिएक्शन करिए तो टुवर्ड्स द प्रोडक्ट साइड हो जाएगा, या फिर एक्सोथर्मिक है तो यहाँ पे आप टुवर्ड्स द रिएक्टेंट साइड कर दीजिए, आंसर करेक्ट हो जाएगा. बट प्रेजेंट केसेस में हम इसको क्लोजली लो वॉल्यूम साइड को एक करेक्ट आंसर कंसीडर कर सकते हैं. आगे चलते हैं. फॉर एन आइडियल गैस डेल्टा H by delta P is. As a matter of fact, one should know, always remember that for an ideal gas, delta U by delta P is equal to delta H by delta P, which is further equal to 0. So इस केस में आंसर होगा 0, that means option A. ये बिल्कुल एकदम स्ट्रेट फॉरवर्ड स्टेटमेंट है जो आप सबको पता होगा, लोअर केमिस्ट्री क्लासेस में आपने पढ़ा भी होगा. विच इज मोस्ट एसिडिक? फिनॉल, बेंजाइल अल्कोहल, मेटाक्लोरोफिनॉल और साइक्लोहेक्सेनॉल. अगर आप मेटाक्लोरोफिनॉल को देखें तो वो फिनॉल से ज्यादा एसिडिक होगा बिकॉज ऑफ द इंडक्टिव इफेक्ट ऑफ क्लोरीन. देयर इज एन इलेक्ट्रोनेगेटिव एलिमेंट इज देयर which is increasing the possibility that phenol molecule is able to release proton much faster in comparison to the original phenol, and hence the option has to be C. Always remember the presence of a nearby electronegative element always cause the more acidic nature of a given compound, and in the present case the C has to be the correct answer. If uranium-235 is bombarded with a neutron, the atom can split into. As we know that U-235 को अगर हम बॉम्बार्ड करेंगे न्यूट्रॉन से तो इसको न्यूक्लियर फिशन कहते हैं, और न्यूक्लियर फिशन में कई सारे ऑप्शंस हो सकते हैं. इन द प्रेजेंट केस अगर हम देखें तो एक पॉसिबिलिटी निकलती है कि हम बेरियम और क्रिप्टॉन का, तो आप देखिए यहाँ पे बेरियम और क्रिप्टॉन का एक कॉम्बिनेशन बनता है, बट यहाँ पे दिख रहा है सी ऑप्शन में बेरियम एंड ज़ीनॉन, तो ये भी गलत है. ज़ीनॉन और स्ट्रोंशियम का एक कॉम्बिनेशन बनता है जो कि फिर से गलत हो गया ए ऑप्शन, क्योंकि स्ट्रोंशियम को दे रखा है लेड के साथ. एक लैंथेनम और ब्रोमीन का सिस्टम बनता है जो कि दे ही नहीं रखा, और कैडमियम एंड क्रिप्टॉन का इसलिए नहीं होगा क्योंकि कैडमियम कभी भी नहीं बन सकता क्रिप्टॉन के साथ बनेगा बेरियम. सीजियम एंड रुबिडियम का एक कॉम्बिनेशन बनता है विच इज ऑप्शन 4, और जो कि लास्ट में करेक्ट भी होगा. आगे द बॉन्ड दैट डिटरमाइंस द सेकेंडरी स्ट्रक्चर ऑफ अ प्रोटीन. बड़ा सिंपल सा क्वेश्चन है. आपको पता होगा दैट प्राइमरी स्ट्रक्चर of any protein is because that amino acid is coupled together through the peptide backbone, which is a comparatively denatured structure. Life has not been started with this structure. Actually, in proteins, the life starts when any protein, the primary structures start taking the secondary conformations, and which is due to the hydrogen bonding between the carbonyl oxygen and the hydrogen of the nitrogen of the peptide backbone. Remember, in the secondary structure, there is no contribution made by the side chain components, and that's where the present case, the C option has to be the correct answer. Hydrolytic reaction of fats with caustic soda is known as. बड़ा सिंपल सा क्वेश्चन है, लोअर क्लासेस में हमने पढ़ा है that fats अगर उसको caustic soda से रिएक्शन कराया जाए तो उस प्रोसेस को हम saponification कहते हैं, which is used in making soaps. So is the present case, the D option has to be correct, which is saponification. Which of the following solution has highest boiling point? In the present case, if you see, there are different types of components have been given, different salts have been given, and ये पूछा गया है कि किसका highest boiling point होगा. As you know in the colligative properties that boiling point, it is dependent upon the number of moles. If you see carefully, in the present case, the C option, it has 4 different types of ions which could be formed, while in the other cases, less number of ionic species are formed, while the molarity of all the species are same. Hence, the C option is very close to the correct, that is 0.1 molar AlCl3, and hence option C has to be the correct. इसी के साथ दोस्तों हम सम अप करते हैं इन क्वेश्चंस को. क्वेश्चन नंबर 51 से लेकर 75 अगले वीडियो में और अपलोड होने वाले हैं. ऐसे ही अपना प्यार बनाए रखिएगा. सब्सक्राइब करिए अगर आपने अभी तक चैनल सब्सक्राइब नहीं किया है, और बेल का आइकन को हिट करिए जिससे कि हमारे आने वाले सारे वीडियोस के अपडेट आपको मिलते रहें. जल्दी ही हम नई सीरीज के साथ IIT JAM की भी आने वाले हैं. इसी तरह अपना प्यार बनाए रखिएगा. धन्यवाद.